Hemophilia A, a genetic condition caused by a change in the factor VIII gene, which is used to produce the coagulation factor VIII protein. This protein functions within blood clotting. Those afflicted experience further inhibition towards their ability to clot. However, there's always a treatment for every genetic condition in one way or another, and this condition happens to be treated with antihemophilic factor VIII. This medication temporarily allows the user to experience normal levels of clotting. What if, however, I told you that there are certain haplotypes of the gene which causes those patients to develop anti-drug antibodies? These anti-drug antibodies attack the drug as it enters the body, rendering treatment useless. Thus, this puts the patient at a greater risk. Hello, my name is Rhys Watson, and welcome to my Flash Talk. I am a second-year student at Mount Allison University, and I am here today to talk about an article by Shankar Guri and his team. This article looks at the polymorphisms in the F8 gene as potential risk factors for the development of anti-drug antibodies during the treatment of factor VIII. Before I continue, however, I should get some vocab out of the way. SNP stands for single nucleotide polymorphisms and is associated with genetic variation among individuals. Continuing on to the article, F8 genes occur in 6 haplotypes, denoted H1 to H6. Generally, H3 and H4 experience the development of anti-drug antibodies more. This experiment was conducted to answer the question, why is this so? And with this information could possibly be used to develop medication in the future which would act against the formation of these anti-drug antibodies. White blood cells response is essential for the development of anti-drug antibodies, and such that response requires the presentation of the peptides by the MHC class II. Experiments conducted measured the binding and half-life of MHC2 complexes using synthetic peptide regions on factor VIII protein where NS-SNPs occurred. Computational binding predictor analysis confirmed that peptides that have wild-type sequence at positions where polymorphisms associated with H3, H4, and H5 bind MHC2 proteins significantly more than a negative control group. These results suggest that wild-type peptides from polymorphic regions of F8 constitute potential T-cell epitopes, thus could explain the increased incidence of anti-drug antibodies in hemophilia A patients with H3 and H4 haplotypes.